Well, welcome everyone. Thank you all for braving the cold. No, my watch, it says -7 degrees. I'm calibrated Celsius. It makes me feel colder. Well, welcome all for our one of our signature awards of the department, the Jeremy Taylor Outstanding Research Mentor Award. And we're delighted to have Tianxi Cai here, Dr. Tianxi Cai from Harvard. We have Jeremy here. So we'll get things underway. So, without further ado, I'll hand it over to Jian for a brief introduction. So the agenda will be a brief introduction from Jian about the award and it's what it's intended for. And we have Sushi, who's a postdoc mentee of Dr. Cai, who's going to give an introduction to her. And then we'll get to hear Tianxi's talk on durable AI in healthcare generalizable learning from imperfect EHR data. And the reception will follow afterwards, right? I'll hand it over to Jian to kick off things here.
So yeah, I prepare very long kind of introduction to Jeremy, but it seems Jeremy came late, so I will be short. Yeah. So it's my great pleasure to present Jeremy Taylor Outstanding Research Mentor Award. That Jeremy has shaped this department in profound ways through the scholarship, his leadership, and most importantly through his mentorship. And so I think we all know Jeremy is an international leader in biostatistics and cancer research, and his contribution have influenced many generations of statistical methodology and biomedical sciences. But what I want to emphasize here is that Jeremy has been deeply committed to Michigan biostatistics as a community, investing time, energy, and care into building programs, mentoring trainees, and supporting junior faculty. These slides briefly highlight the trajectory of academic excellence of Jeremy, and from Cambridge to Berkeley to UCLA and ultimately to Michigan. And what stand out to me is not just the longevity of his career, but the consistent of impact over more than 4 decades. At every stage, Jeremy has been doing work that matters statistically and scientifically. Since coming to Michigan in 1998, he has been central to development of cancer biostatistics here, helping to building up work programs in infrastructure and continue support of both research and training today. Many of us in this department have benefited directly from that foundation. And Jeremy's research contributions are both fundamental and wide-ranging. Many of the methods are now taken for granted, especially in survival analysis, current model, and joint modeling, were shaped in important ways by his work. I think this photo is AI synthetic. Yeah. What I always admire Jeremy's insistence with the methodology should connect with real scientific questions. His work has been deeply embedded in cancer research clinical trial and has had a real influence on practice. So, of course, this award is about a mentorship, and this is where Jeremy's impact and is perhaps most visible. He has mentored dozens of PhD students, postdoc fellows, and junior faculty, and many of them are now leaders in academia, industry, and government. And what really defines Jeremy's as a mentor is how he mentors with high standard and intellectual honesty and a deep respect for his training as an independent thinker. So he invested deeply in his trainee, give them intellectual space to grow and support them long after they graduate. And many of us here have seen this firsthand as students, a collaborator, and a colleague. So the meaning of this award, honors the commitment to training development, excellence in interdisciplinary research mentoring, and lasting impact through people, not only publications. So today we celebrate this legacy by recognizing an outstanding mentor who embodies these values. That's a 2025 recipient, Professor Tianxi Cai from Harvard University. Yeah, congratulations, very well deserved honor. And I will invite Vera and Jeremy to hand the plaque to Tianxi.
Congratulations, it's your award. All right. I have a photo.
So next, I will invite Xu, Tianxi, the former postdoc, and give a more detailed introduction to Tianxi. Okay.
Thank you. Hi, everyone. Thank you for the opportunity to talk about my postdoc advisor, Dr. Tianxi Cai. I will start with a brief introduction about Tianxi's journey. I rarely say this, but Tianxi is an actual genius. And this is why. So she got her bachelor in math from University of Science and Technology in 1995, her PhD in biostat from Harvard in 1999. What's crazy is she was only 22 when she got her PhD from Harvard. When I was 22, I just entered my PhD program, and I thought that was early enough. So this is really amazing. And what's even more amazing is that she's actually the first woman to ever earn tenure in her department at Harvard. So she spent about 2 years at University of Washington and then decided she's going to return to Harvard Biostat. And she went through the whole process of assistant, associate, full professor. And you're only tenured until you're a full professor at Harvard. And she was the very first woman to achieve that. So that really speaks to her, academic achievements. She's currently a named professor and with appointments across multiple departments. She has contributed a lot to our field. She served on many committees, nationally, internationally. She's AE for at least 6 leading biostatistical journals, and she's currently one of the editors of JASA Theory and Methods. She has also organized and chaired many conferences, workshops, sessions. She's reviewed for many journals and grad review panels. Now, about her academic impact. She has published over 300 papers, and she has a really nice balance between methodology papers and scientific collaboration papers. So about half and half, and she's been cited for about 20,000 times. Many of her papers are published in top biostat journals and also top medical journals such as Nature Science, Lancet, and JAMA family journals. She has given too many keynotes, seminars, and talks that I don't have the patience to count them, so I wrote numerous. What really stand out are her scientific and academic impacts. When I did my PhD in 2012, EHR was not a popular topic at all. And at that time, I looked into the literature. The only papers I could find in statistics and biostatistics are almost all from Tianxi. And she really built the statistical foundations of phenotyping and semi-supervised learning many, many years before EHR was even an area. And I think it's fair to say she really opened up this area and made it become one of our mainstream areas. So she's a true pioneer in statistical methods for EHR. She also recently founded Harvard's Translational Data Science Center for a Learning Health System to advance health data science and learning health systems. Many of the tools provided by the center are actually used by people. And I can give you an example. So I work with FDA, and I know that for many, many years, grants after grants, projects after projects, people are trying to implement some of her EHR phenotyping methods because they're really helpful for active post-market safety surveillance of medical products. They are very efficient and reliable, and they don't require chart review. So that's just one example that demonstrated her real-world impact that I know of, and I'm sure there are many of them. This award is about mentorship, so I'm going to talk a little bit about mentorship. First, starting with my own personal experience. Tianxi might not know this. She's actually the reason that I came to academia, that I pursued a career in academia. So when I was in my 5th year in PhD, I was deciding between a few really good job offers in industry and a few really good job offers in academia. But because they're both good, I can't decide. So I called my friend. My friend said, okay, if you can't decide, then think of a role model and then follow their path. And I thought of Tianxi. So I decided I'm going to do a postdoc at Harvard. And even more luckily, later I realized, oh, I can actually be a postdoc of her. We wrote a paper together during my postdoc. And I learned the most important thing in my career from writing that paper with her. That is, she taught me, if you want to do good work, you must solve a real-world problem rather than developing methods for the sake of developing methods. And it's actually very hard because almost always, the real-world problems needs you to step out of your comfort zone. So whenever I run into a failure or challenge, I think of what she said to me and push myself a little bit more. So back to the slides, what's on her CV that you can see is that she has mentored more than 40 PhD students and postdocs, and many of them are now tenured faculty and leaders across academia, government, and industry. But what she didn't list on her CV are the following. Number one, she has provided lifelong mentorship well beyond graduation. Number two, she has made transformational impact on women and underrepresented groups. As an example, she actually founded a summer camp to provide free AI education for high school students from underrepresented groups. She's also the co-PI of the NIH AI/ML Consortium to advance health equity and researcher diversity. So I think it's fair to say that she has really made influences that shaped our field, not just through her papers, but also through every single individual she interacted with. She's always there for her PhD students, postdoc, and even junior faculties and also junior collaborators. So that's, I think, why she should win this award. And here's some pictures of Tianxi and her mentees in the lab. I had a really great experience being a lab member. And as you can see, everybody have a smile on their face, so they're all happy. I would like to end, maybe they pretended, but I don't think so. I would like to end with a few quotes that are collected by one of her former PhD student, Dr. Laila Perez, who's currently an associate professor from the University of Texas, Austin. She said, she never tried to steer me toward academia. Instead, she emphasized that happiness in one's career is what matters most. She encouraged me to explore an internship at a pharmaceutical company. I quickly realized that I did not enjoy a traditional desk job. I disliked being told what to do, and I missed research. When I returned and told Tianxi, she smiled and said she had expected this outcome, exactly why she suggested the internship. Her guidance was thoughtful, supportive, and genuinely focused on helping me find the path that was right for me. And the next quote from her was something that I think I strongly agree with. Tianxi has no interest in developing a new statistical method just for the sake of academic contributions. She wants to change the world. She wants to make our lives, our healthcare better. And that is exactly what makes her an exceptional human being. I have another quote from Dr. Seinert, who is currently an associate professor from Ohio State University. I will also say that Tianxi always nourished her mentees as whole people. She helped us professionally, but also built friendships with us. Because of the time limit, I'm going to just talk about the highlighted parts. Another quote from Dr. Gronsbell, assistant professor from the University of Toronto. I still go to her when I have an issue, and she's always willing to connect, even while she's walking her dog, whose name is Luna, because I also had a conversation with her when she was walking her dog on Friday night. She's certainly a role model to me and to many other students who came from her lab and courses. I find myself modeling the same principles she instilled in me. Invest deeply in young researchers, foster their independence, and lift them up with kindness. And that's all I want to say. Thank you.
First of all, thank you, Shu, for your very kind introduction. If there is a hole, I want to go into it right now. I do have a minor correction. Actually, Harvard did have many wonderful female professors tenured in our department. But I was fortunate enough that I think I was also a PhD student from the same department. I do feel that I grew up in that department. And I have to also say to you and many of my mentees, how wonderful you guys are. But if there's anything you see from me, I also want to say that I truly benefit from my own mentors who have always been so generous. Like I cried in front of them. I had so many issues with my life, with many things, and they gave so much to me. So I feel extremely, extremely fortunate. So first of all, again, thank you so much for this amazing honor. It's really humbling to be here at Michigan to receive such a wonderful award. For many of us, Jeremy has really been a role model that balance, achieve scientific contribution, but at the same time, in his mind, he's really thinking about how to help other people to achieve their dreams, not only just to make him a better researcher, and to be recognized in his name is truly something I will cherish forever. Looking back at my career, I would say the most rewarding part of this journey is, in fact, the wonderful people I get to work with. One thing great about Harvard and similar at Michigan is how amazing our students and colleagues are. So we all benefit so much from them. Some of you asked me what's my mentoring style. The truth is that I simply enjoy sharing my life with them. That's why they know my dog's name is Luna. I have a young son, he's 10, and he went to my lab many times. He went to my lab meeting. I just really, truly enjoy sharing a life with them. But I really view my students as my partners, sometimes partners in crime. And in many ways, they feel like my grown children, except that I didn't have to feed them. I didn't have to worry getting up at night to feed them, and they don't talk back. That's a major difference. My son always talks back. But in all seriousness, they truly are the heartbeat of my work. Whether we are, working on an algorithm or paper revision, they really make me a better scientist. They have pushed me to be a better person. To my mentees, I really thank you for letting me be part of your lives. You have shaped not only my career, my heart, and this is more than any award could give. So in the spirit of collective discovery, I like to share some of the research that I work together with my students. So as Shu mentioned, in the past, I would say a decade or actually almost two now, I've been focusing a lot on the analysis of electronic health record data. And over time, my interest grew, and I work a lot on cross-institutional, cross-language. And of course, as of today, there is obviously a rise of AI, right? It's in front of us everywhere. You cannot even avoid it. With this in mind, I want to talk a little bit about some of the methods that we are working on, really to think hard about how we can empower EHR for discovery that can help us to improve healthcare research, or improve the delivery of healthcare. But I would first like to acknowledge that for most of the work we do, it's truly a teamwork. Many of them would involve a team of physicians, computer scientists, my students, and here I'm just listing many of also my collaborators that have made the work possible. So there is a lot of talk about AI. And I have to say that I think it will, I still think the bubble will burst. However, even if it bursts, it fundamentally have changed and it will continue to change both our lives and our research. And in healthcare, this is an analysis done by a consulting firm trying to estimate, how much value there is with AI in healthcare. If you look at the category that list in this circle, it's actually already happening. For example, the first thing you see is imaging. Imaging is probably most advanced. That there have been a lot of successful tools actually implemented in healthcare that at least help us to make a better diagnosis or prognosis. The second category is drug discovery. So drug discovery actually is another amazing area that I never thought that I would be able to do. But because of my students and my postdoc, they are really interested. I learned so much working together with them and understand how we as statisticians can also contribute to actually leveraging both EHR data, public available data, genetic data to help us with drug discovery. And obviously you can think about, another like a clinical decision support tools, etc. Back to EHR, like why is EHR even a thing that matters in healthcare? Of course, that is the data that's closest to patient, right? Because if you implement, for example, the next thing will be it's in the healthcare system that will directly communicate at point of care. But also it's just huge amount of data that captures so much of wide range of phenotypic information that include codified information, so like ICD billing code, prescription, lab result. We also have a huge amount of data embedded in clinical concepts. You can extract via natural language processing. I'll dive a little bit deeper later. And of course, there's imaging. You can also have imaging that's linked with radiology report. And that's one amazing thing about EHR. For every imaging, there is a report. So you can pair them, and that will allow us to basically take an image, even generate a report automatically, or from the report, you can also generate image. So this is a relatively mature field, I would say, compared to many other aspects. But for research, in many institutions, including Mass General Brigham in Boston and also VA, they also link EHR data with additional data sources like a disease registry, where you would collect additional outcome information that's not routinely collected as part of healthcare, such as disease activity for rheumatoid arthritis. That's not something that they actually document clearly, say your disease activity is 3.2. So that's typically curated through registry, and you can link that data with the actual EHR data. For larger institutions like VA, we also run clinical trials. We are currently working on a subset of patients are part of the clinical trial, and the remainder of patients are not part of the trial. We're trying to use both sides of the data to understand, for example, comparing a triple therapy, which is very cheap, $8 a day, versus a few $1,000s of biologics. But the original trial, for example, shown equivalence, but we want to understand, are there certain patients that actually you should use one versus the other? In addition, there are lots of biobanks. I'm sure you are familiar with UK Biobank, and I know Michigan also has a biobank. In Mass General Brigham has a biobank that we have genotype, for example, 60,000 at the VA, the Million Veterans Project in their last year or 2 years ago in the science paper. I think at that point, they have genotype 700,000 patients. So you can see there's a huge amount of information that we can leverage to understand about the disease or the biological, for example, causes of the disease. So I want to give you a few examples. What do we actually use EHR data for? So in collaboration with my psychiatrist, we have a center grant on suicide prevention. So we developed a risk prediction model, and this is right into the heart of Jeremy's work in like survival analysis with longitudinal data. For example, at any given moment, like this is a clinical scenario. Let's say that, for a patient who have already been admitted to psych ED, emergency department, you look at their history up to now, and you have to predict whether someone might commit suicide in the next 3 months or 6 months. So we looked at psych ED because the general population, actually, the prevalence is too low. So even you see the AUC looks amazing. But actually, if you look at their positive predictive value, like it's very low. So basically, it's not clinically meaningful to take any action. But in psych ED, the base risk is very high already. They're already admitted to emergency department because of psychiatric condition. So among those patients, we actually look at the top 5%. Their positive predictive value was 50%. They have 50% of chance of committing suicide. So this is the subpopulation we are trying to understand. Should we provide this information to the treating physician, the family, and we leave them to decide what to do? And we're running a randomized trial. The other group is routine care. We don't provide this information. So all we want to know is it helpful for them to receive such information? And we are currently recruiting patients. So EHR is also longitudinal. So one of the most valuable information we have is actually longitudinal treatment information. So for most of the clinical trial, like it's, they usually like a randomized patient, and usually at best you can do like a ITT or doing causal if they switch. But typically you cannot have very long follow-up. You don't know what should happen to them after they fail this drug. So that's a question we can answer in EHR. So we actually looked at the longitudinal sequence of medication that the patient took, and then we just simply clustered them to understand in the real world, how are the patients with rheumatoid arthritis actually managed? So these are the 5 clusters that we came up. But what makes it really interesting is that this is actually a joint clustering between the VA and Mass General. We never share patient-level data. You can develop algorithms to jointly cluster these patients. And the interesting part really is that the treatment patterns after clustering are very similar. We just allow the hospital to have different percentage of patients in each of the cluster, but assume that they should share, similar clusters. Basically, within each of the cluster, what are the treatment patterns? You can see they actually do look very similar. So with the treatment cluster, we then look at the genetics of these patients, because we always wonder, we being my rheumatologist friend, she always wonder whether or not there is some reason for why these patients in the actually left 3 panels, they all start from TNF, and then they had to switch to some other drug. The very right one are TNF persisters. So they might start with one TNF, but they might switch to another TNF, but they respond to TNF. So the middle 2 actually are the ones that they have multiple switches. And we looked at the different clusters and look at the genetic profile of these patients. For example, the genetic risk score for rheumatoid arthritis and a few other rheumatic conditions and autoimmune disease conditions. And we did find that the TNF persisters look different from the people that actually don't respond to the treatment. So you can see that this is actually one of the really helpful ways for us to have biobank linked EHR data, because we have longitudinal information. But EHR data, take a step back. One of the main reasons it's very powerful is that it has huge amount of information. You have every diagnosis, all the prescriptions, lab results, etc., as well as the narrative clinical terms that appear in clinical notes. So we actually try to summarize the information from the EHR and try to build a knowledge graph. So at high level, for those of you who don't work on knowledge graph, at least one aspect of knowledge graph is we derive a representation for each of the node. So each of the entity, let's say rheumatoid arthritis, it has its own numerical vector of embedding to represent rheumatoid arthritis. Then if I take rheumatoid arthritis as a target node, I can look at around all the other features, what are the top clinical entities that are very close to the vector of rheumatoid arthritis. So we basically developed an algorithm to both learn the representation and a statistical testing procedure to test whether or not the link is there. So that is one of the things I enjoy working with statisticians. So we can actually analyze the data to understand how we actually distinguish, noise of being linked versus the actual signal. So we develop a testing procedure to identify basically which of the links actually exist among the entities. And this is actually one of the tools that we developed that the VA has a digital library, and they use that, they absorb that into their library that VA researchers can go and search. Basically, if you start your project, you don't know which feature should you use, because EHR is a little bit too overwhelming. They could have millions, and this knowledge graph, we looked at 60,000 entities. So if you have a project on rheumatoid arthritis, you don't want to start with 60,000. As statistician, we all know you quickly will have, basically the dimensionality is too high. And this will help you to actually narrow down to the sort of the entities that you should pay more attention to. We developed a tool like the one on the right-hand side. Actually, you can come to our website. You can say, okay, this is a disease I want to study. What is the feature list that should I use? And you can click, you can download. So this is actually a tool that we now use with all our collaborators who work on the EHR. But from the representation side, we have basically a numerical vector to represent the semantic meaning of each of the code. You can link it back to the patient. So if the patient has, a list of these codes, you can map the representation of the code back to the patient to come up with a patient representation. So here, as a proof of concept, we looked at all patients who had their first diagnosis of mental health disorder. So any of the mental health disorder, we look at that first diagnosis. We look at 1 year since their first diagnosis, basically early, beginning journey of their mental health disorder, and then using our representation learning to cluster patient. And this cluster into 5 different clusters. And you can see that the word cloud here basically tell you within each of the cluster, what are the entities that's driving this cluster. And then we plotted their risk of suicide attempt and suicide ideation. You'll see there are 2 clusters that actually are very different from the rest of the clusters, which is cluster number 2 is depression predominant, and also cluster 4 is psychosis dominant. Actually, there was a recent paper, when we were just wrapping up this paper, there was a recent clinical paper suggesting that these are the 2 major risk factors, basically schizophrenia or major depression disorder. Of course, this is not entirely surprising, but we just want to show as a proof of concept, how can we go from the large scale EHR data itself, learn the knowledge and go back to the EHR so that I can discover more information from the EHR. But in addition to clinical science, my former postdoc, Jun, who just joined NBC-UAI as a junior faculty, he has deep interest in bioinformatics. I know nothing about bioinformatics. So when he said his interest, I sure, let's learn about it. But I really enjoyed the journey because he really pushed me to really understand, because we had discussion, he ran into problems. But we did two amazing projects together and also changed my view of what statistician can do. So the first project we started with basically taking chemical structure of a drug. And there are many embedding models. You can turn that into an embedding. Basically, take a chemical structure and you can embed, have a representation for the drug. And then we link that with many, many different kinds of data. But one data that we have uniquely are actually data derived summary level data from electronic health records. Those data will tell us what drugs are related to what diseases and what diseases are related to each other. So the representation I showed you earlier, and that is further linked with additional resources from FDA. You can go to the FDA website. You can identify what are the potential reported side effects of the existing drug, what are the drugs that are actually their approved indication. So from there, you can train a model to predict a new drug that you haven't seen, a chemical structure. You only have the chemical structure. Can you predict what's the indication? So that was the first time I realized, oh, that's really cool. So sort of gradually pushed me towards this direction. In the second project, we actually looked at basically, Harvard is part of this so-called undiagnosed disease network. And typically, they're undiagnosed because they have some rare condition, and some of which actually have a condition that we don't even know that they existed. So through this collective effort over the years, they have been able to diagnose many of the patients. But usually for rare diseases, they tend to have missense variant. And as you know, we have alpha missense. So what alpha missense can do is that you take a missense variant, it can predict how likely it's a pathogenic, but it doesn't tell you what disease does it cause. So that's what we want to do. We want to take a missense variant. We want to use alpha missense to say, okay, these are the pathogenic variants, and can you predict what disease can it cause? So through this, we actually use a protein language model to compare proteins that are wild type protein with this mutation, and we're trying to learn like what is that aspects of the protein that actually make it cause what disease. So again, we actually showed that this can potentially predict what the potential cause of the disease. We went back to the undiagnosed disease network in the past few years where they were able to successfully diagnose the disease and show that you can indeed find signals. I wouldn't say that I will rely on this particular tool to just automatically diagnose the disease, but it definitely can help you to narrow down what are the potential disease. You can also, of course, look at the symptoms of the disease, and whichever the biologic implication and the clinical implication match each other, that's the most likely disease. So through this effort, I realized that as soon as we know how to sort of get the information, where the signal comes from, why, like the training of the algorithm can learn the signal, and which part of the signal, which part of the data did the signal come from. But I, again, really want to emphasize that without Jun, I would never work on that because I can't. I even failed high school biology, so I think my biology career ended then. But with students' help, I suddenly feel much more powerful. But I painted a rosy picture, right? All these seem amazing. Like there's so much information, so much we can do. But at the same time, EHR data is very noisy. Like there's so much noisy coding for example, at Mass General, among patients with at least 3 diagnostic code of rheumatoid arthritis, they're 58%. Only 58% of them actually have RA. That's 3 codes. So you can see how much noise there is. And also codes tend to be very susceptible to data shift. Like across system, you might think that they might be using different codes. And across over time, there is like ICD-9 code to ICD-10 code. I won't dive into details of this because you actually have a local expert, Shu, here. And also Jessica Grunsbell, she also worked a lot on basically overcoming the noisy coding. Recently, with the rise of GPT or large language model, we also want to empower ourselves to get more information from EHR so that we can take that information to enable downstream analysis to have access to more high quality data. Because this is really helpful to us, because to get a very accurate information from the EHR, you need like chart review, but that's obviously not scalable. If you want to work on, tens of thousands of patients, it's not feasible. And unfortunately, for some of the more complex disease phenotypes, such as disease activity that I will go over in a bit, traditional machine learning cannot do very well. Like the highest AUC we can achieve is about 0.78. And the transportability, by the way, is horrible. We trained a model at Mass General. When I transported to the VA, the AUC went from 0.8 to 0.6, and vice versa. We still haven't figured out why. But at the same time, if you send detailed information to a language model and ask them to read, they do a reasonable job. So we want to leverage language model to basically help us to annotate the data, and so that we can basically have a semi-automated chart review system that can scale, that can run on tens of thousands of patients. But if I have time, hopefully, I will also talk about, how can we actually improve the durability of AI models. So we talked about, how to empower EHR, the quality, improving the quality of the EHR, also make your model more sturdy in the sense that more transportable. But at the same time, the system or the patient cohort naturally change over time. As they change, these algorithms tend to decay. So basically, they tend to degrade. How can we actually, when we build the algorithm, already have that in mind, plan for future to make the training more robust to this kind of degradation? So through the help of my many amazing students and postdocs, we developed a large language model-based information extraction system that essentially can read the clinical text, identify what is a clinical entity, identify what is an assertion status, is a family history, is a negation. And if it's a lab result, for example, ejection fraction, we need it for subtyping heart failure. But ejection fraction is actually only in the notes in echo report. So you actually want to send the LM to recognize the value of the ejection fraction, so you can assign them as like HFpEF, HFrEF, different subtypes. So we developed essentially an information extraction and structuring agent. We actually developed two different agents. One is like you can plug in any LM to it. It will run, and it will basically, you can, for example, the clients that I mentioned here, you can plug in Llama 3, you can plug in GPT-4o, you can also plug in like the smaller DeepSeek model. So you can see that it actually, even with a reasonably small 32B model, it can achieve a fairly high accuracy. But we are too poor. Even for that, I can't afford it, both in time, because we don't have enough GPU. And also, if I have to use commercial GPU, I literally cannot afford it. It's too expensive. So we also fine-tuned a Llama, I think it's a 7B model called a Genie. Actually, it can specifically even extract value information very accurately. That can scale reasonably well. So at this stage, I would say that, even though I'm a statistician, but because I have a fearless postdoc who was willing, like Shen here, actually, he's the person really changed my perspective, because he actually developed the first NLP software for our group before I even know exactly what's NLP. But ever since, I feel like it freed us that we can get data ourselves. We can even help other people to get data. Then you are actually sitting, on the same table instead of like being a helper in the backyard. So this actually, now we are trying to deploy both at all the institutions that we work with to help them. For example, particularly for rare diseases, a lot of the information is in the notes. So we use that to extract the information that we need. But for more complex phenotypes, such as the disease activity, I mentioned a few times already, That's basically something that's unique for rheumatoid arthritis. It involves like, how many swollen joints? Do you have like a joint pain? And is there a lot of inflammation? So there are basically multiple aspects that the disease activity score involves. But clinical notes is not so detailed. It actually cannot recover the actual clinical score that involves so many aspects because they have sometimes very vague information. So we actually deployed an LLM agent. All I want is just the score, then I'm going off to do my causal modeling. And I thought that this is great, right? ChatGPT is so good now. Why don't we just do it? I told my student, let's just do it in a month. Then it turns out it's so hard. So the first time they finished, you can see that the agent's initial accuracy is about 80%. So I was not very happy because it took us so much time. And then we realized that actually, even at that point, human made so many mistakes too. So I suddenly don't feel as bad, right? We're almost 50-50. Then we also recognize that there's actually a lot of ambiguity in the notes. We need a domain expert to explain to us, if you see conflicting information, what does it mean? Which one I need to value more? So that's when we realize that you really need a so-called human agent co-learning. So we work together, like the agent point out, you guys made a mistake. And then we also learned from the human input to say, okay, let's correct our understanding of how the phenotype is defined. In the end, the agent accuracy was about 90%. And it's actually comparable to this is actually even an earlier iteration. After we further refine, it was actually a bit higher than 90%. So this is where we decided it's good enough. So when it's good enough, again, my goal is not here. I just want the agent so that it can extract data for us. But because of that, it greatly freed us in terms of sample size, right? I wanted thousands, not like 100 people. But this will greatly improve our ability to do downstream, for example, causal modeling. So I want to give you one example of how we can leverage LLM for real-world evidence. So as you know, many of you here, including Shu and many experts here, do causal modeling. So in causal modeling, of course, we all know there's confounding bias, right? Here, I'm even not assuming I'm not worried about unmeasured confounder. But I also have issue like disease activity not directly available, but now I have an agent. But despite my best effort, my agent is only 90% accurate. But one thing I think none of us, including myself, earlier to recognize how bad the medication code was. I thought medication prescription, how can it be wrong? Do you know why it's wrong? So you really need to work on it before you can know. So apparently, sometimes a physician will give you the prescription first before you ask for insurance. And for there's another complication because it's a biologics. There is actually serious side effects. So they usually do certain testing on the patient first. So they will give them the prescription at the same time ask for insurance approval or ask for this testing. In the end, they might abandon that plan. But the code is there. It's not going to be canceled. So that's why not only the drug information is wrong, but sometimes the patient, you are studying drug A versus B. You'll think, okay, maybe there is some, A misclassified as B. Apparently, there's also a C. They're taking a completely different drug. So if you're comparing A to B, the C patients are not even eligible for your study. So that's another layer of complexity. But fortunately, this is somewhere like semi-supervised learning can help us. But because there's a complication in the noise in the label, so basically we leverage both still the human chart review we view as a gold standard, and like the final adjudicated chart review, it's a gold standard, which is still small amount, but I have a large amount of LM annotation that's highly accurate. So here, I just want to mention that we developed a semi-supervised causal estimation that account for both the noise in the outcome, but also noise in your treatment assignment group. So we propose a semi-supervised framework. The basic framework is actually really standard. You can see that many of you probably have seen something like this, right? In semi-parametric inference, you basically write down the influence function, efficient influence function, then estimate it. So this is a very similar tool, except that in our case, it's a bit more complex because of the complexity in the noise in both A and the Y. And in our current paper, as I mentioned, I also further correct for the so-called ineligible patient. They are not even they shouldn't be part of your study. So here I want to show you the result that we find from basically comparing two treatment groups for rheumatoid arthritis patient. And this is actually my favorite pictures that I found years ago because Renoir is my favorite artist. And you notice that actually the picture on the right, his hand, actually he has severe rheumatoid arthritis. Unfortunately, at that point, they didn't have good enough treatment. So there's a lot of like bone erosion on the right. And in the end, he actually couldn't paint anymore. He did a sculpture because he had to wrap his hand to avoid the pain. But here, back to the RA example, we actually looked at, if you look at the timeline of patient with different treatment over time, so you can see that there's basically after, in more recent years, after the initial treatment is given, for example, the typical one is you start with methotrexate. If it doesn't work, they'll start you with anti-TNF. So it's a very standard. So when that happens, you cannot do causal. For example, you can never do causal start with methotrexate versus TNF because there is almost no randomness in the assignment. But after you fail the first TNF, then physicians don't know what's the best thing that they should do for the patient. So that's the thing they want to find out. Should they try another TNF versus try a different mechanism? So this is basically the causal question we want to answer. And we have about 2,000 patients in the EHR that received a prescription of TNF, a new TNF, or switching to a different mechanism after failed the first TNF. But here, about 24% of them were not on either of the treatment. So you can see that it's a very dangerous thing to do because it's a substantial percent of patient. But here we also develop an agent to infer treatment information over time. And here, I just want to take a quick look at the third one, the green one in the middle. So this is basically using our method, using the LM outcome information as well as the treatment information. The last one is actually, if you don't use LM to annotate the Y, you use traditional like neural network or XGBoost. So you can see how much wider the confidence interval is. So basically, this is showing you that despite your best effort to use traditional machine learning to phenotype, it still cannot do well because it's such a complex phenotype that it's largely contained in a lot of the description of the text. So the last part that I want to talk about is algorithm degradation. So why is it important? So here you can look at the growth of AI as a medical device. So if there's a new algorithm now, it's approved as a medical device. So that's how they are treated. And you can see the approval number almost exponentially increased. But if you do a little bit of literature search, there's a much focus now about the degradation. So they tend to degrade after deployment. As you can imagine, right, over time, many things can change. There could be coding change, like she worked on ICD-9 to 10. So if you develop a model during the ICD-9 era to ICD-10 era, it could be very bad. But patient population generally change. And also across system, right? When we develop such a tool, it takes so much effort, so much resource. At best, you can probably test in 1 or 2 external. But by deployment stage, you want to deploy across, if you're a company, right, you want to deploy as much as possible. So then the shift is between healthcare system, right? How they're coding, like the coding practice might be different, or even just how they're treated, they're managed are very different. So how can we actually train a model with this in mind, knowing that your future target population might be different from any of the population that you have in your hand? So here is an actual real study that we did. So I mentioned that we developed a suicide risk prediction model. So this is to say, okay, I want to validate my suicide prediction model in 2019 on the left and in 2020 on the right. But in 2020, that was our COVID year. So I even separate into first half year, second half year, because the second half year we got vaccine. So you can see that even between first half year, second half year, they're drastically different. And then if you look at the one on the left, basically the more on the right, they're closer to the year of the validation. So this is essentially showing the so-called aging of machine learning model. So the older model on the left did much more poorly compared to the more recent model. So that's to say, how can we sort of avoid or slow the aging of machine learning model so that when we deploy to a different system, they can actually perform equally well. And this is another example where we actually simply were looking at polygenic risk score. And as you all know, that genetics among all of us, it depends on our race, ethnicity, it depends on many other aspects. And here, if we developed a genetic risk model for UK Biobank, which is predominantly white, and we validate it on other population, and the y-axis is basically the error bar. You can see that when you deploy on the all-white population, it performed poorly even within UK Biobank on Asian particularly and other mixed population. And if you look at MGB, it's even more horrible because MGB is a completely different health system and different population. So you can see that the external sort of validity of the polygenic risk score from UK Biobank really is very poor based on at MGB. So the question is, can we build a polygenic risk score that can be generalizable to a range of external populations? They might have different percentage of different kinds of population. So this is basically asking for a paradigm shift. Don't just look at your own training data or validation data. You should already design for future. Basically, think beyond what we have in front of you. So instead of optimizing a model for a single historical data or two historical data, we want to optimize the worst case performance on future a collection of populations that you can imagine that you have information on. So essentially, the key idea actually comes from the literature of so-called group distributional robust optimization. They create a so-called uncertainty set. Essentially, these are the collection of populations and collection of possible future models. And you want to build a model that cannot do poorly in any of these distributions. So just to be a bit more detailed, if you look at here, you have L different source populations. They each have their own model. And you have a target population that might have a different distribution. So Q is the conditional distribution of Y given X for the target. And then the source population is P of Y given X indexed by L. You have a capital L total population. So the group DRO essentially try to optimize the worst case scenario within this so-called uncertainty set. What's an uncertainty set? It's creating a collection of population using the observed source population. So we imagine that my future might look like some kind of mixture of the current source. So that's my so-called uncertainty set. I don't know which one. And then you optimize the worst case performance. So basically the worst accuracy is optimized. So by doing so, you achieve the so-called robustness, right? Just by definition, it basically is trying to bring this so-called adversarial into the consideration. And the one problem of this approach is that, for example, there's a very well-known paper by Peter Buhlmann and Nikolai Meinhausen, where they look at a linear model and they can show that actually the solution essentially is your original, the target, sorry, the collection of betas, you form a convex set. It's a projection from 0 to that collection set. So they actually use 0 as a benchmark as your null model. So essentially, there is a very nice geometric interpretation. However, that approach can be very conservative for any particular target population. So what we consider is that what if I have a collection of past historical data, but I'm standing in 2019. I have a tiny bit of target data. And my goal is still not to optimize for 2019, but I'm optimizing for future populations that are more similar to 2019. So that way I'm guiding my existing source populations or source models to say, if the too old of a source model that does poorly in my observed target population, I don't even want it. I want to weigh them down. So that way, essentially, you are making your so-called uncertainty set much smaller, and that will overcome the conservatism. So we use this actually for example, in the suicide example, I just want to give you here an example here. The X is like any kind of feature map. So you can have pre-trained embedding from transformers, or you can just have raw X as your raw observed data. And our goal is to say I have a collection of source models and I have a small amount of target data. And we train an algorithm to essentially minimize the, to find the beta that, the optimal beta the best beta such that my worst gain from an initial estimator is optimized. So again, back to the min max or max min scenario to protect our conservativeness. And the one last thing I want to mention is that in the so-called group distributional robust optimization literature, there was very little consideration about uncertainty. I have L models. You never told me which one of your L models has more training data. What if one of the models have like 100 observations? It's highly unreliable. And you let that completely change, right? Their impact to your final estimate might be too large. So we also try to bring the uncertainty into the picture. And also after I build the model, I also want to know how much uncertainty is there in my estimated model. But I want to point out that this is actually an irregular inference in the sense that it's not normal. So you don't have the asymptotic normality because you are basically on the boundary of your parameter space. But you can use a resampling approach to actually find a union of the confidence intervals to cover the truth. Just to show you in the real data, like here in the suicide prediction example, so the x-axis is actually different target year, and our model is adapted, the red one on the top. So you can see that when I want to validate, for example, in 2020, like for our model, even though before 2015, our model also did worse compared to after 2015, but it did much better compared to other models. Do you guys know what's the significance of 2015? Why this happened? That's when we changed from ICD-9 to ICD-10. So there's a huge shift to your underlying model. So you can see from here that it's very clearly shown there is a bump And here on the right-hand side, I'm just plotting the so-called aging effect. So the slope here is just showing you how quickly your model is aging. So you can see that our model is much, much more stable compared to the purple curve is actually the standard approach. At any given moment, I take all past the data, I pull them all together and estimate. So that's only fair, right? We all have access to same amount of information. And you can see that by doing DRO, you can achieve a lot more stability. And the last part I mentioned that, we also account for the uncertainty. And here the outer circle is from our estimate. And you can see the last lines. I just want to mention that, if you look at across all the genetic markers, we want to basically build a model to estimate what the weights that we should use to construct a polygenic risk score, but we also want a p-value, like a significance, and also the confidence interval. So you can see that we can capture a lot more significant variance. We did a lot better in terms of external validation, but at the same time, our confidence interval is much, much narrower. So this is just to show us that sometimes we can learn from the heterogeneity. If you observe the data, the source models already have a lot of heterogeneity. You should already think about it. Don't think about it like I take the most recent or take the average or take the most stable. Essentially, this is teaching us like how to construct the data. Essentially is teaching us how to look across different models to understand what signals are basically most invariant. So intuitively, essentially, when there's a lot of instability in the signal, you might be better off not to use it. So just to wrap up, I would say that, if you think of validation of AI algorithm as medical device, it requires us a lot more thinking about so-called cross-environment testing, because the unfortunate truth is that the failure of such a model is silent. You don't even know that it failed. And health systems don't have the ability, or it's just too costly, right, for us to constantly reevaluate the model. So I think having this mindset of building the model in a more robust way will be helpful. But I also want to point out that as much as I love the LLMs, we are actually working on several projects on the adaptive sampling of which of the patient and which of the nodes we should sample to get the most expensive model versus the cheapest model. Because I really think that unless we see a huge gain in using the more capable model, the cost is huge. Like many people are complaining just about the power, the energy consumption, right? So I definitely don't, as much as I want to embrace the AI part, but I also want to be very conscious to only really get it when it's actually available, it's actually helping us. And the last part I didn't have time to talk about is actually when you think about multi-institution collaboration, there's a lot more complexity due to, for example, different coding system, different language, and also how do we integrate preclinical data and multimodality from protein, genes, imaging. And I really think that it's an exciting field for all of us at this moment, even though I don't think I want to abandon my statistical principle. And I want to also thank my students and my mentees again. Without them, this would not be possible. I really feel like I'm their main cheerleaders, but I learned so much from many of you, including Shu here. But thank you all.
So the question and answer. So comments first. So thank you very much for coming. It's been a dazzling array of material you presented. I think mind blowing to sort of see how you understand how you can do so much. And as Shu said, I'd always thought of you as a genius. So just sort of related to what you talked about at the end, right? So we teach the statistical methods, which is regression and probabilizable and Bayesian stuff, but you're large language models, right? So there's a monumental amount of complexity underlying all these things. So sort of three questions maybe related to all that. So sometimes you meet resistance when you collaborate with people. It's too complicated. I can't really understand it. So do you use that sort of attitude? And then what's the role for this standard errors and logistic regression and all this? Where does it come into play?
So that's a great question. So first of all, at least for students, I feel like when the first wave of AI came up, like you asked me what's the relevance, for example, logistic regression versus like large language model, I feel like I don't think I can think critically without the knowledge that I have acquired through, for example, my understanding of longitudinal data survival. I actually had this paper that I'm so proud of because my clinician friends insist that they are going to take the patient who died, take a back, look at a month, and then compare to patient who didn't die. They don't know how to match them. They want to match some kind of case control. And then they want to claim how important the marker is. I had a long debate. I was just arguing with them. I said, you can't do that. You don't have all patients aligned right a month before in your office, right? Patients come randomly to your office. They might die in 5 months. They might die in a day, right? So in that sense, that's why prediction of survival is so hard. But I feel like without that, I don't think I feel that I can make meaningful contributions. But at the same time, if you look at the losses, like of all these fancy models, the last layer is just a logistic regression. They just add layer by layer by layer, right? You just like aggregate. It's almost like our model aggregation. They just have different way of aggregating. So I agree with you. I actually don't have a good answer. How should we teach our students? Like they must feel very stressed. There's logistic regression I need to learn. There's survival analysis, the asymptotic inference. But at the same time, there's like large language model, all these like, how can you accommodate all of that? But I think I like to do both top down and bottom up together. Like I still learn my logistic regression. Many of my students still work on theoretical work. But at the same time, I enjoy learning. So I also have collaborators, like I have CS postdocs. So I really encourage us to go out of our comfort zone, just work on it, learn from them. But I really think that if I didn't, I really actually appreciate all the papers. Many of them are written by you. Like I still feel that I can think a bit more. So for example, we work on surrogate marker, right? This is actually really interesting. So I have AI as an agent. You can think of my LM agent as my surrogate marker. I can do causal modeling. So I can actually quantify to what extent, if I want to estimate the treatment effect on my disease activity, is reflected or captured by if I directly use AI-derived label. So that's actually an aim that we wrote for one of our FDA grants. Unfortunately, it got canceled.
Questions here?
Thank you very much for the excellent talk. And I especially very impressed by your effort to construct a knowledge graph using EHR data. And so the EHR data, I think there is a lot of dynamic and temporal data, and how I would like to know that using those data to relate into the knowledge graph.
Yeah, excellent question. So there are two kinds of temporality in the EHR data. One timeline is a calendar time, right? As you know, different drugs became available over different time. So from that end, we are actually constructing sort of evolving knowledge graph. How did our knowledge change over time? So that's like a high level temporality. Patient also have temporality. That's actually one of the major advantage of EHR, that you know what happened first, then what happened. For example, for a drug, right? If it's a side effect, it tends to happen after, right? If a drug is used to treat the disease, the side effect happened after the drug, but the indication happened before, diagnosis happened before the drug. So that's exactly why temporality matters. So we actually built another knowledge graph. It's a directed knowledge graph. Basically, it has a direction. Like what happens first, what happens, basically who's on the left, who's on the right. And we show that by using directed embedding, it actually can help you more for predicting a downstream task when it's like predicting a future event. So definitely temporality matters in this case.
Did you publish that research?
Yeah. So it's called the DOME. It's just an algorithm that we developed to look at directed embedding.
Yeah, Jeremy, you talked about investigators that might have resistance to AI because it's too complex. I'm in exactly the opposite situation where I've got some investigators think that AI is going to solve every problem and that you don't need a statistician because they're going to go and we should give them the data all the time. We're going to go and give them ChatGPT so that they can go and say, what's the best predictive model for Healy or doing diabetic foot ulcers? And we really don't need the statistician. And so I don't want to be defensive in the sense about, I'm getting close to retirement, so but I sorry about it. I'll be fine. But I don't think it's right. And so I've heard you talk about some of the reservations. Have you met this reaction? And how do you deal with it without it sounding like you're trying to protect your profession as a statistician?
So first, let's think about what is AI, right? Everything can be called AI. So they claim, everybody claim, right? So in a way, like I hate to chase the fashion, but I would say in my mind, like a lot of the training principles, for example, they do contrastive learning. If you look at the loss, it's exactly matched case control. So if you look at matched case control, the loss is exactly in their contrastive learning. But somehow suddenly became computer scientists field. And like I asked them, do you know what's a matched case control? They don't. So I would say like on the one hand, I also want to defend because I'm a statistician too, right? But on the other hand, I feel like I don't, how should I say? I view myself more like a data scientist. I don't want to only wear the hat of statistician. So basically, at the one hand, part of me, whenever when I talk to physician, like I wear both hats, so that I don't think I need to defend. They just find me valuable. But over time, I actually find many clinicians still really enjoy working with statisticians because the computer scientists, I don't want to stereotype, but they're too optimistic. And sometimes they can't interpret the result back to the clinician. So I think if we acquire the skill of both, like using our traditional logit regression and transformer, whatever it is, we can also show them, actually, you have not much to gain. So basically, it depends on signal to noise ratio. It's just a fundamental principle, right? The model complexity versus how much data you have. So I don't think one always dominates the other, but I feel like we should, occupy part of the AI field too. Why not us?
Question, we have a reception coming up. Sorry, we just wrote. Question.
Yes, thank you. Yeah, for wonderful talk. So I think, yeah, I'm interested in you talk about the, like when you have ICD 9 to ICD 10, so the coding certain changes, right? So and then you talk about the data is missing, and then you even have a lot of coding differences. Even people put coding like the rheumatoid arthritis, they even have 3 different codes. So a lot of things there. Actually, I'm interested in how you view it. How can we really support the data integration? Have integrated certain people talk about the federated learning using OMOP or many other systems. How you view? How can we put really all that together to support the better AI analysis?
Yeah, that's a really great point. I feel like I still spend almost 90% of my time trying to resolve data issues. But at the same time, it makes me realize how important that step is. So actually, I would say at least half of my current research is indeed trying to automate or guide the data harmonization across system, across language. So we built like these representation learning algorithms that can simultaneously embed all like a shared common ontology as well as your local code across many different systems, including different languages. So that if you imagine in the future, you are at a particular health system, but you have some kind of summary level data you already shared with us, you can actually use that as a smart Google search. Like you say, I want to study rheumatoid arthritis, it will show up all the codes that belong to your system so that you will not miss that. This is so painful. Like we had the drugs. So there are many like infusion drugs. They're actually coded as procedure. They're not coded as Rx norm, which is the standardized code for medication. So we look at the VA, they have nobody treated with infliximab, which is the most frequently used anti-TNF for IBD patients. It's like, that's impossible. And then we dig deeper, realize that VA didn't code them as Rx norm. It's a procedure code. So that's horrifying, right? If you do your research and you say, okay, this is the, the number of patients using this, and you realize, oh, I didn't use that drug because it's a different code. So definitely this is one of our focus right now. Like, can I use relatively simple summary data from the EHR to automate, at least help me to check, like, is there some code that I'm missing? And also, how do you actually interoperate, right? I'm right now establishing a collaboration with University of Geneva. They speak French. So that's like, has always been my dream. I want to work on actually both, for example, English version of the clinical notes and the French version. So this will help us to basically establish this kind of interoperability.